In the previous lecture, we discussed the organization of the nervous system. So we talked about peripheral and central nervous system, automatic and somatic nervous systems. Now we'll discuss the basics of a neuron. Now again, this should be reviewed just like the last lecture, but just in case it isn't, we're going to go over it. So here are the outcomes for this lecture. There are 4 of them. Now when it comes to a neuron, there are 3 main parts. The cell body, which is also referred to as the soma, the dendrites, and the axon. Now the cell body is shown here by this circle. And this is where the nucleus, which is this little green thing in the middle, whoops, man, I keep hitting buttons there, sorry about that. So is where that nucleus is located. Now the cell body also helps to maintain the structure of the cell, and just like all other cells, the cell body and nucleus is where protein synthesis occurs. Next, we have the dendrites, which are shown by this circle. Now, the dendrites are the primary site where information is received from other cells. Once a dendrite receives information, it sends this information along to the cell body where this information is processed, and then something happens, or in some cases, nothing happens, depending on what the cell body decides. Finally, we have the axon, which is shown here. Now, the job of the axon is to take information from the cell body and pass it along to other cells in the form of action potentials and then ultimately neurotransmitter release. Because these action potentials will eventually reach the axon terminal, which is the end of the axon. And in the axon terminal, there are these containers called vesicles, which contain or which store neurotransmitter. And when appropriate, that neurotransmitter is released. So the axon is used to send information to other cells with the help of these neurotransmitters. Now, just a quick disclaimer here that the next few slides are the exact same as those that I use in PE 220. Okay, so having said that, I want you to remember it's possible to categorize neurons as myelinated or unmyelinated. Now, an unmyelinated neuron doesn't have any myelin, and myelin is just a type of insulation. It's a fatty layer or a fatty sheath that surrounds some neurons. Now, as a result, in an unmyelinated neuron, when an action potential occurs, each area of the membrane must become depolarized. This causes the adjacent areas to become depolarized, which causes another adjacent area to become depolarized, etc., until the end of the axon or axon terminal is reached. Okay, now this is a relatively slow process, okay? And signals or action potentials travel at about 1 meter per second along an unmyelinated neuron, okay? And in many cases, that's completely fine, but in other cases, that's way too slow. Just think if you were standing on something that was hot with your big toe. I'm about 2 meters tall. That means it would take 2 seconds for the signal to get from my big toe to my brain saying I'm stepping on something hot, and then another 2 seconds to get back down from my brain to my big toe to move my leg. Okay, and so that would be 4 seconds. And by then my toe has burned off. Now, saltatory means to leap. And so in a myelinated neuron, they experience something called saltatory conduction. Or in other words, the action potential leaps, and notice that I've got that in quotes, because the myelin reduces the areas where the ions can move. This means depolarization only occurs at the nodes or the gaps in the myelin. And these gaps are where the voltage-gated channels are located. Now, if you look at this GIF, the neuron on the left shows how a signal travels in an unmyelinated neuron. Notice it travels relatively slowly, while the neuron on the right shows how a signal travels in a myelinated neuron. It essentially jumps between these nodes. So saltatory conduction starts when voltage-gated channels open. This allows a rush of sodium to go into the cell. This sodium then pushes the sodium that's already in the cell towards the axon terminal. The movement of this sodium depolarizes the membrane, which opens voltage-gated sodium channels, which are at the next node. Now, while it would be a ridiculously fast process if the entire neuron was myelinated, that's not possible. And the reason being is that the reason we need nodes is because the sodium that goes into the cell can only travel so far. Because of the gradient, there's only so much energy. And so it has enough energy to push sodium down to the next node, which will open more channels, which will let more sodium in. Okay, saltatory conduction is really fast. Okay, and as opposed to the 1 meter per second in an unmyelinated neuron, in a myelinated neuron, this can be about 100 meters per second. So after this little lecture, I hope that you're able to do the following. Identify the main parts of a neuron, describe the function of each part of the neuron, and explain how saltatory conduction occurs.